Hey everyone, welcome back for another lesson. So today I'm going to give you an introduction to protein synthesis. So far, previously we have learned that genetic information is stored in the chromosome, and then this genetic information is transmitted to daughter cells through the process known as DNA replication, and the information from here is then expressed through transcription into RNA. In this case here it is mRNA, and then eventually mRNA is then translated into proteins. So the pathway of this protein synthesis from mRNA is called translation, and it is called translation because it is very similar to translating something from one language to another. So in this case here we are actually translating the language of nucleotide sequence into the language of amino acid sequence, and then this amino acids are joined together by peptide bond to form proteins. So let's talk about this genetic code. And what is genetic code? So genetic code is basically a sequence of nucleotide bases in a gene which eventually determines the sequence of amino acids in a protein. And they are nothing but they are just a group of 3 nucleotide bases. They are called as triplet in mRNA, which code for an amino acid. So the question is, why there are group of 3 nucleotide bases? Why can't there be just a pair of nucleotides which can represent as genetic code? So to clarify that further, suppose here we have 4 nucleotide bases. These are the general 4 nucleotide bases. So here we have adenine, cytosine, guanine, and uracil. Because this is RNA, so this is it's uracil. If it is DNA, it would be thymine instead of uracil. So suppose if we were to make a pair of nucleotides, so which would be called as double coat bases. So here we have 4 nucleotide and we want to make pair of nucleotides. So that would be 4 to the power of 2, and it would give us 16 possible different codes. However, the only problem is there are 20 amino acids, so the 16 different codes would be insufficient to code for all these 20 amino acids. Therefore, the double code basis is not possible or is insufficient to code 20 amino acids. On the other hand, if you take a triplet code basis, which is the 3 nucleotide bases in current situation, so we would actually have 4 to the power of 3, which would make us 64 different possible codes, and that is actually more than enough to code 20 amino acids. Therefore we have triplet and does not have doublet. I don't know if it is called doublet, but I'm just calling it as a doublet. So therefore we have triplet and not the double code basis. So let's look at a little bit differently. So this is, suppose we have DNA strand, and I have actually break them apart, considering that these are the triplets. And suppose consider this is our template strand, and this template strand is then transcribed into RNA through the process of transcription, and so here we have mRNA, and you can see here that these are the triplets. So these triplets eventually, the group of triplets then eventually called as codons, and then these codons are then being translated into amino acids. So these are the amino acid sequence. So this one triplet is then translated into one particular amino acid, and once these amino acids are coded, they are then eventually joined together by this peptide bond to synthesize protein. So this language or this information of nucleotide bases, which is then translated into the language of or the sequence of nucleotide bases is translated into sequence of amino acid. That process is called translation to synthesize protein. So now let's talk about how this genetic code is being translated into amino acid sequence. So suppose here we have one codon. This is called AUG, and one thing to remember that these codons are always read from 5' to 3' direction on mRNA. And suppose these are the bases, so this is the first base, this is the second base, and this is the third base, and the genetic information is translated through this genetic code table. This is also called as RNA codon table. So here this is the first position. Consider this is also the first position. So this first position is also called as 5' base because this position is located close to the 5' end on mRNA. And the middle letter here, the middle nucleotide base, is the second which is located on the second position. It is also called as middle base. And the last base, which is the third position, is called as 3' base because it is located on the it is located close to the 3' end on mRNA. So the way the genetic information is translated, we first need to understand this RNA codon table. So now let's focus on this 4 rows which contain 16 amino acids, and for all these 16 amino acids, their first nucleotide base is adenine. Similarly, for this 16 amino acids, their first nucleotide base is cytosine, and similarly for this at the bottom, this 16 amino acids, their first nucleotide base is guanine. So this is not very difficult to understand. Now second, let's focus on this first column. So this also contains 16 amino acids, and similarly all these 16 amino acids, their middle base is uracil. And likewise, if you focus on the second column, all these 16 amino acids located on the second column, their middle base would be cytosine. So likewise it would be for adenine and guanine. Now let's see how the third base is determined, last base is determined. So if we focus on amino acids of these 4 different rows, so these 4 separated rows which again contain 16 amino acid whose last codon would be cytosine. And if you see uracil, so uracil, if you see uracil, which is highlighted in red, so for all these 16 amino acids, their last base would be uracil. So suppose let's take an example here. So we have here AUG triplet, right? So if we take A, U, and G, so this is methionine. So AUG codes for methionine. So you can see here that A, U, and G. And it is also important to mention that methionine, the triplet AUG is also it also signifies translation initiation. So it codes for translation initiation, and it also codes for methionine. So this is so to understand this RNA codon table is very important, so that you can anytime translate the genetic code into amino acid sequence. So let's just quickly do some practice. Suppose here we have mRNA, and here we have codons. So these are the triplets, and combination of these triplets are codons. So let's now decode these codons to determine their amino acid sequence. So first we have UCA. So if we go here, the first base, which is this U, so U, then C, and A, so that is serine. So this is serine. Now let's focus on this GGU, And then always remember, you are reading from 5 prime to 3 prime, so we always read from here to here, not from 3 prime to 5 prime. So the second one is GGU. So G, then middle base is G, and last base is U, so that is glycine. Now once again, UCA. So the first base is U, second base is C, and the last base is A. So that is serine. And last one is UAC. So here we have first base U, second base, the middle base is A, and the third base is C. So that is tyrosine. So this is not very difficult, guys. You can just take any codon sequence and and then try to determine the amino acid sequence from there. So once we determine this amino acid sequence, these amino acids are eventually going to join together through peptide bond to form proteins. And this information which is translating the information of nucleotide sequence into amino acid is called translation, because we are translating from the language of nucleotide sequence into amino acid sequence. So now let's talk about some of the important features that characterize genetic code. So first thing we need to know about genetic code is, like we already learned before, that it's a group of 3 nucleotide bases which code for 1 amino acids. In this case, suppose we saw that the codon AUG, which code for methionine. Now second, this genetic code is translated into proteins in a non-overlapping manner. And what I mean by that is, suppose here we have nucleotide bases, and these are from 5 prime to 3 prime end, and this is our first triplet, and suppose this is our second triplet. And as you can see here that this two nucleotide bases is already overlapping between first triplet and the second triplet. Likewise, if we have third triplet, you can see that there is an overlap of nucleotide bases between first, second, and third triplet. And suppose if we have 4 nucleotide bases, four triplets, you can see that there is like so much of overlapping of just one nucleotide base between this first, second, third, and fourth triplets in this series of codons. So this is called overlapping manner. Now in non-overlapping means you start from the codons, they are read from 5 prime to 3 prime end. So if you start from here, this is the first triplet, and then this is the second triplet, third triplet, fourth and fifth, and you see that there is no overlapping of nucleotide bases between these codons, and therefore it is called as therefore it should be in a non-overlapping manner. It should be read continuously, not in an overlapping manner. Now third, the code is read from a fixed starting point as a continuous sequence without any punctuation or without any comma. And what I mean by that is, suppose here we have nucleotide base, and suppose this is our first triplet, and then you miss the second you miss this nucleotide base, and then this is our second triplet. So this is called comma, because you miss we are missing the one nucleotide base here. Now likewise, we have third triplet, and then this nucleotide base was missed, and then this nucleotide base was missed, and this is the fourth triplet. So this is called punctuated code, because there is a comma in between. The nucleotide bases are missing, and therefore it is called punctuated code. The ideal way it should be done is just read continuously from 5 prime to 3 prime end without missing any nucleotide base, or in a commaless manner. Therefore it is called commaless, and it's also called as continuous code. So first thing you need to remember that this codon is translated in a non-overlapping manner and in a commaless manner. Now, third, codons have meaning. What I mean by that is we saw before that there are 64 codons, and out of 64, 61 codons code for amino acids, and 3 codons, they do not code for any amino acid. And why? Because suppose if you see here that there are 3 codons which is indicated here by stop. So these codons, they do not really code for anything. They do not code for amino acid. In fact, they indicate the translation termination. So therefore there are 61 codons which code for amino acids, and there are 3 codons which does not code for amino acid. Therefore they indicate translation termination. So here the remaining 3 codons, which are also called as nonsense codons, they serve as termination or stop codon, and then the example of this codon is UAA. So U, A, and A. So this is UAA, then UAG. So U, A, and G, and UGA. UGA. So these 3 codons, they indicate translation termination, and they do not code for any amino acid. That is important to remember. Now, fourth, genetic code is degenerated. Degenerated means each amino acid is coded by several codons. So suppose if you see here leucine, so leucine is coded by 4 codons. So C U U, then C U C, C U A. So therefore leucine is coded by 4 codons. Likewise alanine, valine, they are all coded by 4 codons, and therefore they are called as degenerated, except for methionine. So methionine, there is only 1 codon for methionine as well as for tryptophan. So there is an exception, so that is methionine and tryptophan, which only has 1 codon which code for this particular amino acid. Now this genetic code is unambiguous or specific, meaning that all these 61 codons, they only code for 1 amino acids. So suppose if we have methionine, so it is AUG, it only codes for methionine. Suppose if we have leucine here, CUG only code for leucine. It is not going to code for any other amino acid. So each 61 codons encodes for only 1 amino acid, and degenerated means each amino acid is coded by several codons. So hope you understand this. It's a little bit confusing, but it is also at the same time it is also very simple to understand. And the last is genetic code is universal. Universal means its specificity has been conserved from very early stages of evolution, and it is virtually universal across all organisms such as archaea, bacteria, and eukaryotes. However, only in one condition, where there is an exception in mitochondria in which few codons have meaning different than it is shown here in this codon table. So that is the only exception we have, that in mitochondria some of the meaning of the codon is different than specified here in this codon table. And lastly, I just want to mention that any alteration in this nucleotide sequence in mRNA may result in an incorrect amino acid which is being inserted into polypeptide chain, and that could be potentially causing any disease or even death of any organism, and that could be lethal. So and then that is called mutation. So I'm definitely going to, it's a big topic, so I'm definitely going to cover that in a separate video. So thanks so much for watching the video, guys, and I really hope that you learned something from this video. And I will see you soon in the next session, next series of protein synthesis. Thank you so much. And if you like the video, please like and share the video and subscribe the channel. Thanks so much.